Good evening, everyone. If you would make your way to the seats, we're about to start the program. That's just me. You can sit there. Where? If they are watching. Wow, that actually worked. So good evening. It's so wonderful to see you here this evening, many familiar faces and many new faces. I'm Susan L., Vice President and Executive Director of the Foundation for Barnes-Jewish Hospital. So happy you could join us this evening. This series, this Leading Edge Medicine series that we have always excites me. There's always something new, innovative, transformative, and discoveries that we can talk about, and tonight will not disappoint, I promise. It's an exclusive opportunity to get up close and personal with physician scientists who are truly on the leading edge, hence the name of the series. Because you've joined us this evening, I'm guessing that you share my interest and excitement as well. Anyway, I'm hoping you feel similarly. This is a new venue for us, so I hope you like it. I'd like to say thank you to our servers, to View 17 and the Foundation team for making such wonderful arrangements. Next time, maybe it'll be a clear day that we can see out across the beautiful city. But thank you for the service, excuse me. And we will have more reception after the program, after we have the presentation, so that, if we run out of time during the presentation, there will be ample time after to visit with one another and with our presenters. Also, a note, we are recording this evening, so you should be aware of that. As I'm looking at these lights, I can't see you. I mean, I'm right here, but I can't see you, but I do see the lights. When donors make a philanthropic investment to medical research, the return is ultimately better patient care. The two physician scientists who we're going to hear from tonight, Drs. Holtzman and Milbrandt, are world-renowned specialists in their fields. And the research that they do together is a testament to the special collaborative culture that we are so privileged to benefit from and enjoy here at Barnes-Jewish Hospital and Washington University School of Medicine. It really is a unique working milieu that you probably won't find. I've been told by the physicians and scientists that it's really unique and very special to be here, not only in St. Louis, but at Barnes-Jewish and Washington University School of Medicine, where there is this interplay and interchange of information and education and working together and collaborating. Excuse me. We at the Foundation are really privileged to be part of the fabric, of that institutional fabric. The Foundation helps donors enrich lives, save lives, and transform patient care through charitable gifts. And you are changing lives whenever you make a gift through the Foundation, whether you direct your philanthropy to Barnes-Jewish Hospital, Barnes-Jewish West County Hospital, Siteman Cancer Center, Goldfarb School of Nursing at Barnes-Jewish College, BJC Home Care and Hospice, and Evelyn's House, or Washington University School of Medicine, as we're about to hear tonight. It's my privilege to recognize our Foundation board members who are here. You will recognize them by them wearing the silver badges, and our chairman of the board, Dick Miles. And I guess now, was it Ed Sullivan that used to say, it's going to be a really big show? What is it he used to say? So we have a really big show, and I'm really privileged to introduce and happy to introduce Bob Cannon, president of Barnes-Jewish Hospital, group president of BJC Healthcare, who is going to introduce our featured speakers. Thanks.
Thank you, Susan. Good evening, everyone, and really a hearty thank you for being here this evening. There are a great many things that make great medical centers great, and one of them that's right there at the top is the support of the donors. And all that you do, your generosity and the philanthropy that you provide allows us to do so much more than we could otherwise do. And just getting going for the evening here, before I privileged Dr. Holtzman and I were speaking a little bit and talking about a physician scientist colleague of ours who got started early on with a grant, and that led to a very productive career, and now to an appointment as a new department chair. And the work that the foundation does and that you do as donors, and we've talked about this before, provides a lot of that early research activity that allows our scientists, clinicians here to be so much more competitive in seeking extramural funding from the National Institutes of Health and others, that allows us to really maintain ourselves as one of the great academic medical centers of the world. So I just wanted to take a minute to recognize you all for the role you play, and to thank you again for being here this evening. Last year, the foundation was privileged to award over $15 million in funding to the kind of research efforts at the Washington University School of Medicine that result in many of the advancements that we will hear about tonight. So I don't want to delay this much longer because we really are in for a treat. The speakers that we have this evening, their combined expertise in really groundbreaking research in neurodegenerative diseases and the development of new treatments, the collaboration that Susan referenced to tackle neurological diseases in a way that were previously untreatable is really amazing, and there are many more treatments on the horizon. A new dawn of medicine is, I think, on the horizon now, and our speakers this evening, I think, will give you some real insight into what that means. So first, Dr. David Holtzman is the Andrew B. and Gretchen P. Jones Professor and Chair of the Department of Neurology. He's Scientific Director of the Hope Center for Neurological Disorders and Associate Director of the Knight Alzheimer's Disease Research Center at the Washington University School of Medicine. Dr. Milbrandt, also with us tonight, is the James S. Mcdonald Professor and Head of the Department of Genetics, Executive Director of the Mcdonald Genome Institute, Co-Director of the Needleman Center for Neurometabolism and Axial Therapeutics, as well as the Professor of Pathology. And Jeff, I should get some credit for getting through all that, so there we go. Professor of Pathology, Immunology, Medicine, and Neurology, all at the Washington University School of Medicine. So our speakers this evening have been very generous and gracious, and wanted to let all of you know to please feel free to interject with your questions as they move through their presentation. We very much want this to be interactive and meet your needs this evening. And so I think without any further introduction, we'll just welcome Drs. Milbrandt and Holtzman to the podium, to the table here, and get on with our evening. Gentlemen, thank you.
I'm going to let Dave go first, but a couple things to start with. So you should know before we start that Dave and I have been not only colleagues, but we've been really good friends for a really, really long time. So if Dave were to say something unflattering about what we do during the presentations, it's all good. It won't cause any trouble. So I just want you to know that you probably all know the joke is that a neurologist is really good at telling you what's wrong with you. Now, the part about fixing that and helping you get over that, not so much. So tonight, we're here to tell you a little bit about how we're rectifying that really this is a new dawn where cures for neurological disorders are well on the horizon. So it's not just about diagnosis anymore and telling you what's wrong, but it's also how I can help you. So, Dave?
Well, great. Well, Jeff, I think that's what's so exciting about being here tonight is that it always has been said that neurologists basically diagnose things, but they don't fix anything. And I think what we'll be illustrating tonight is the fact that we really do think there's already some examples where there's some great treatments that have been developed, but even more on the horizon. I think before I get started, though, as Jeff said, I've known Jeff for a while. I know he looks maybe younger than I am, but 30 years ago, when I was still in California, I'm originally from St. Louis, but I was in California, and I was trying to collaborate on a project, and I called up Jeff because I knew that some of the areas he was working on were related to what I was interested in at the time. And as soon as I found out that he was from Nebraska, which is where my mother was from, I think we immediately bonded. So it's great to be able to speak at the same time as Jeff, which I don't think we've ever done before. He's such a good friend. So let me move on to why we're here. So what I'm going to talk about is disorders of the central nervous system, so the brain and the spinal cord, and when this part of the nervous system degenerates. So just to put everybody on the same page, in our brain we have nerve cells, as illustrated in this slide. But most of the nerve cell is not what's called the cell body, but this long process called the axon. So if you think a nerve cell might be in the brain, and it's a process that it sends out to communicate to other cells, goes all the way down, in some cases, like 3 feet, all the way down to your spinal cord. So that illustrates the axon, and here's the cell body. What happens in neurodegenerative diseases is that both of these things can degenerate. Sometimes the cell body, or often the axon, and Jeff's going to talk a lot more about the axon. So the cells can sit in the brain and send their processes out, for example, to the spinal cord, or a cell could sit in the spinal cord and send its process out to the muscle where it could work. So we'll talk about diseases that affect all of these structures. So you've probably, some of the most common impactful diseases of the central nervous system are listed here. So I'm sure you've all heard of these, and some of the famous people that have developed these, such as Lou Gehrig, who developed ALS, or amyotrophic lateral sclerosis, Ronald Reagan, who died of Alzheimer's disease, Woody Guthrie, who died of Huntington's disease, and then Muhammad Ali and Michael J. Fox, who have Parkinson's disease. So one of the things about these disorders is that while they may seem quite different when you see a person who has these problems, there's a lot in common between the disorders. And one of the ways that we've tried to attack that at Washington University and Barnes Jewish Hospital is through what was mentioned earlier, which is through collaboration. While we have fantastic scientists that work on these problems, one of the ways that we can probably get a lot more out of our work is if we work together to work on these diseases. So one of the things that we did a few years ago, gosh, it's already now 2003, so 16 years ago, we formed a center called the Hope Center. And the purpose of the Hope Center is to improve the lives of people living with neurological disorders through collaborative research that aims to discover fundamental mechanisms that lead to neurodegeneration, as well as repair, and then to try to translate these into new understandings and develop cures and treatments. So the way that we've approached this is that over time we've developed collaborations of people that work in the Hope Center. They're dispersed around our campus, but it involves now over 134 faculty from every single department at Washington University, both at the medical school as well as at the Danforth campus. You can see we have about 70 MDs or MD PhDs, 1 dentist, 34 junior faculty. And if you include all of the staff and graduate students and postdoctoral fellows that work in these laboratories, there's over 675 people in these labs. And I think in doing this, it's really built a great place where if you don't know how to do something, you go to another lab and they often know, and it's really led to a lot of additional discoveries, I think, that we would not have come to otherwise. So a lot of these disorders, as I said, cause different kinds of problems. I'm just going to illustrate I'll talk a little bit about Alzheimer's disease just as an example of some of the problems that we're dealing with and ultimately how to develop treatments for them. So to make sure everybody's on the same page, dementia is a decline in memory and other aspects of thinking that is sufficient to impair your social and occupational functioning. You can see that the prevalence of dementia is only about 2% at age 70, But by age 85 and over, it's about 50% of people develop some kind of dementia. So Alzheimer's disease is a specific disease that causes dementia, and in the United States, it contributes to about 60 to 70% of people who become demented. There's a variety of other diseases that can also cause dementia as well. For example, there's diseases called Lewy body dementia, frontotemporal dementia. And if in aggregate right now, just Alzheimer's disease itself is been estimated in several articles to be costing the United States economy about $250 billion a year. And right now, if you add up all these other disorders, other neurological disorders that are like it that I showed you earlier, it's estimated that it's costing the US economy about $1 billion a year. So these are a huge problem. And right now, we really don't have great treatments for these. I'll show you some of the promise that's coming forward right now. So in the case of Alzheimer's disease specifically, a few years ago it was estimated there are a little more than 5 million people with dementia from Alzheimer's disease in the US. And if we develop no way to delay or prevent the disease, by 2050 it's predicted this number will almost triple. So this is why there's so much effort going into research to try to develop better ways to treat this, but also the other diseases that we'll discuss as well. So one of the main findings that's come out over the last 25 years, which is one of the main reasons I actually came to St. Louis at this time, is the realization that these diseases don't start when you develop symptoms. So for example, if somebody is developing Alzheimer's disease and they start showing evidence of memory loss, often the first sign of the disease. On this graph, it shows that if somebody died at the stage of very mild dementia due to Alzheimer's disease, the two fundamental pathological features of the disease in the brain that lead to the problem that we see clinically are the development of these things called amyloid plaques and neurofibrillary tangles. So two different proteins the body makes. Normally they're produced and they're cleared away, but when they accumulate in the brain, they cause a lot of problem. So what you can see is if somebody dies with very mild dementia due to Alzheimer's disease, the amount of these amyloid plaques that accumulate in the brain has already reached its almost maximal extent. These neurofibrillary tangles, which I'll talk about more in a moment, which seem to cause the actual damage in the brain that we visualize, are not at its full extent, but they're on their way up already. And so the same principle is true for most of the other neurodegenerative diseases. So for example, we'll talk in a moment about ALS, or motor neuron disease. In that disease, there are other proteins that accumulate, not these proteins, but different ones. And they also start accumulating in the spinal cord and in part of the brain years before the symptoms start. The same is true in Parkinson's disease, where a protein called alpha-synuclein builds up in the brain in structures called Lewy bodies. Those structures are building up years before people develop a tremor, for example. So the reason this is so important is that if we're going to develop really effective treatments for these diseases, what we're probably going to have to do is detect the disease in this, what we call the preclinical period, when people are still normal, but these things are occurring, and then we can actually administer a treatment to try to delay or prevent the further accumulation of these bad proteins. You could think of this somewhat analogous to somebody who develops, let's say, a heart attack at a particular age or a stroke. That's due to often a cholesterol building up in your arteries, and that started many years before that stroke or heart attack happened, and now you probably all get checked every so often for your cholesterol levels, and if they're too high, you may treat that with a diet or with a statin drug or something like that. I think the same thing is going to happen for all of these diseases, and that's ultimately the way that we will make the biggest impact on trying to prevent or delay these diseases. So this is just an illustration in the case of Alzheimer's disease of some of the pathology that you can actually now see in living people. So I remember I said there's two proteins that build up in the brain. One's called amyloid, the other's called tau. These are some of the progress that's been made in detecting these things in the living brain. So each of these are scans of individual people where a person was injected with a tracer in their blood that gets into the brain, and it binds to these neurofibrillary tau tangles. And what you can see is that these people who are cognitively normal have a little bit of buildup in their brain. But it only starts really building up, this red color here in yellow or red, in people that are actually becoming cognitively impaired. And so this is a huge advance, because if we have treatments that target these proteins and either remove them or delay their appearance, we can now detect who's developing these so that we could monitor these during treatment to try to prevent or delay the disease. So for all of these diseases, there's a variety of publications that suggest that, like in heart disease or cancer, that lifestyle and other factors, which there is evidence that we feel is likely to delay the onset of disease if you do these things. And they're kind of common sense, but they actually have been shown to have effects. So regular exercise, adequate sleep, a heart-healthy diet, not smoking, preventing obesity, and preventing and treating diabetes. Now, these still are being explored more formally, but these are just things that obviously that we all recommend that people try to take care of. But even if you do all these things due to either genetics or age or other factors, it's not going to necessarily prevent you from getting some of these diseases. So this is where we get to the whole point of tonight's presentations, where we're going to talk about the fact that we really do, I think, have some promising treatments in some cases that have been developed for some of these diseases, but in many where we have a lot of evidence now that we really think really effective treatments are on the horizon. So the way the field has approached this so far is, as I said, in these diseases like Alzheimer's, Parkinson's, ALS, et cetera, proteins build up in the brain that should not be building up. And so one of the ways that seems obvious to try to treat this would be to remove these toxic clumped up proteins from the brain or spinal cord, and then to try to decrease their toxicity. So the first example I'll show of this is where people have been developing antibodies the body can make, or we can generate in animals and make it so they're compatible to give to people, and see whether those can be used to get rid of the accumulating toxic protein. So I'll just show you one example from our own work where one of the proteins that I just mentioned, this tau protein that forms neurofibrillary tangles, that is important not only in Alzheimer's disease, but a few other diseases that cause dementia, like frontotemporal dementia, progressive supranuclear palsy. The field has made animal models, just mouse models, where you can put a human gene into a mouse and cause it to get the same kind of changes in the brain that you see in the human disease. So this is an example of a part of a mouse brain where we overexpress this toxic protein tau, and all of this brown stuff is the accumulation of the tau protein in the brain of a mouse. And what we had developed in my lab a few years ago is an antibody to this tau protein, and if we administer it regularly to these mice, you can see that there's much less of this brown stuff accumulating. And while we don't know yet whether this kind of a therapy will work in human beings or not, this particular antibody was actually licensed to a company called C2N Diagnostics, who then worked with a large pharmaceutical company called AbbVie, and this is now in phase 2 clinical trials in humans in Alzheimer's disease, as well as in another disease called progressive supranuclear palsy. So we're hopeful that if you can actually either get rid of or prevent the accumulation of a protein like this, that it might really have an impact on the progression of these diseases. So that's one example. Another example, which has already been showing some clear effects in people, are treatments that either increase survival factors for nerve cells in the brain, or that decrease the production of those toxic proteins I was just showing you. And I'll show you some examples of one thing, and I'll explain what it is in a minute, called antisense oligonucleotides, which have led to what I would argue is probably the first therapy that actually saves people's lives with one of these neurodegenerative diseases. So this is an example of something just published in the last 2 years of a drug that's now called nusinersen. This is what's called an antisense oligonucleotide. And so the disease it was developed for is called spinal muscular atrophy. This is a disease that we often see in newborn babies, where motor neurons that are in the spinal cord that project to muscle begin to degenerate soon after a baby is born. And so it's a disease that looks like ALS, except it happens right out in many people, some in adults, but usually after birth. And basically a baby will be born very floppy, and then usually over the first 6 to 12 months, the baby becomes ventilator dependent and dies, 100%, if you have that form of the disease. So it turns out that the cause of this disease is there's a protein that isn't being produced by the body due to a genetic mutation. But there's another protein that is very similar to it that the body doesn't express at high enough levels to replace the function of the normal protein. So what was figured out is that if you could use one of these, what's called antisense oligonucleotides, which are small pieces of a DNA-like molecule, that you could enable the gene that isn't expressed high enough to work to get it to express again. So what the investigators did through a series of studies, first in animals and then tested in people, they were able to inject this antisense oligonucleotide into the spinal fluid of these babies, or sometimes in adults if they have the same disease, and it turns on this other normal protein that's a survival factor. So again, these kids normally die by the time they're a year of age. And what you can see in this trial where they gave this drug, 100% of these people, of these babies, start as normal, or almost. They don't need a ventilator. And you can see if they're untreated by 26 weeks of life, 40% are on a ventilator. If they're treated with this drug, only 20% have had to go on a ventilator, so 80% are still doing okay. And what's really interesting is that some of these children now that have been treated with this drug are living, they're already 3, 4, 5 years old, and they look like normal kids. Basically, these are kids that would be dead otherwise. It's unbelievable. So this is a real success story that is now being tried with other similar methods for other diseases, and I'll talk about that in a moment. So as I told you just a moment ago, one of the proteins that builds up in these nervous system diseases is tau. So I showed you this antibody that is being developed that hopefully will remove some of this material from the brain and then prevent its toxic effects. Another way to decrease this tau accumulation or other proteins that accumulate is again to use this strategy called antisense oligonucleotides. And one of these, what they can do is to also block the expression or production of a protein that's building up. So in this case, Tim Miller, who's a professor of neurology at Washington University, has been working on both ALS as well as some aspects of Alzheimer's disease recently. And what he did was develop an antisense oligonucleotide to the tau protein. And what he shows is that using the same model I just showed you, in some of these animals, these are mice, they build up this green material, which is, again, the abnormal tau protein. So what he started to do was inject these mice into their central nervous system with this antisense oligonucleotide. And he showed that this suppresses the production of this tau protein. And you can see it mostly blocks the accumulation of the protein in this animal model. So it turns out that this model goes on to develop brain degeneration, and the animals die quite early. But when he does this, it also strongly increases their lifespan. This treatment was just licensed to a large pharmaceutical company called Biogen, and it is in phase 1 clinical trials in Alzheimer's disease. So this is a really promising thing now that we've already seen that this other drug, other antisense oligonucleotide can have a big effect. I think I'm really hopeful that this kind of approach will work in these types of disorders. The other thing I just mentioned that Dr. Miller is working on is ALS. And there's some forms of ALS where a protein called superoxide dismutase, or SOD1, accumulates in the spinal cord and in the brain. So Tim has actually been working on this for over 10 years. And he also developed these antisense oligonucleotides to block the production of this SOD1 protein. So there's both mouse and rat models of this disease where the mice and rats get ALS, and they basically die when they're, depending on the model, in middle age. And so what he did in these experiments is give this antisense oligonucleotide to either mice or to rats. And in this case, it extended the lifespan of these mice for 37 days, which in a human is equivalent to 4 to 5 years. So if you know anything about ALS, you realize that most people that develop this disease die within 2 to 3 years. So to be able to extend the life of somebody who's developing this by this long, this would be a huge win for the field. And maybe if this kind of thing was given before people were symptomatic, it could really maybe even prevent the disease if this kind of approach worked. So this is super exciting, and Tim is very interested in developing an ALS center at Washington University and Barnes-Jewish Hospital, and so this is an area where I think we have a lot of things to come in the next few years. And finally, I'll end before Jeff speaks about treatments that modulate the brain's immune system. And this might be relevant for a lot of these neurodegenerative diseases, and it's already relevant as a treatment in multiple sclerosis. So just to show you an example, in multiple sclerosis, frequently early in the disease over the first few years, you have relapsing and remitting episodes where somebody can get affected and then they get back to close to normal and then they get affected again. And the reason these episodes occur is that cells of the immune system that are in the periphery enter the brain and they attack the central nervous system. So some of the treatments that have been developed over the last 20 years for MS are things that block the entry of these immune cells into the brain. So just to show you one example of an already approved antibody or drug that prevents the entry of these immune cells into the brain is called ocrelizumab. This is a drug, in this case it's produced by the company Genentech and Roche. And so what was shown here in a clinical trial is this is the probability over time that people with MS given a placebo will develop progression of disease. So most of the people have some progression over several years. And in people given this new drug, you can see that their progression is less. Now, it doesn't look so impressive, but in fact, if you actually look at MRI scans of the brain and you look at recurrence of inflammation, it really strongly suppresses that. So it has an effect. This was approved. There's other drugs that have some similar effects that are approved. The big challenge in MS, other than these immune modulating drugs, is to develop things that block the long-term neurodegeneration. And that's something I'm going to turn over the podium to Jeff, because that's something that he has been working on and will tell you more about. So I'll stop there.
Well, thanks, Dave. That was great. And I just wanted to say a few words about how we think about a problem to start with so that you can follow the logic that we've pursued. So most of biomedical research takes into account these things. So either patient observation, it could be an animal observation, clinical phenotype, more and more DNA sequencing. As you know, Washington University has one of the largest sequencing centers in the world, the Mcdonald Genome Institute. And so DNA sequencing takes a large role here as elsewhere, and of course increasingly computation. Once we know about a alteration, a perturbation from normal, we want to define the consequences of that. So that's where you get into molecular mechanistic studies that I'll describe a little bit about how we got to where we do. This is necessary to really develop a drug, to really find out what the target is that you're trying to modulate to fix the defect. That can involve many, many different types of things, genomics, imaging, et cetera. And then once you have that, then of course you want to find an intervention. That could be a drug, that could be gene therapy, it could be oligonucleotides, as Dave mentioned, or increasingly stem cell therapy. And as you know, for cancer, immunotherapy has been very successful in the last few years. So that's kind of the roadmap of the way we think and the way we pursued this problem. So this is my good friend and colleague that shares laboratory space with me in this nice new building, the Couch building at Washington University. And Aaron and I share a distinctive thing that we both spent 4 years in medical school and never saw a patient. And there's a lot of patients that are glad that's probably true. But we thought that the laboratory was a better place for us to try and impact lives, not individually as is done by outstanding physicians, but more maybe globally if we could discover a cure. We worked with Phil and Simon Needleman, who liked the idea that we should study axons, this confluence between neurodegeneration, metabolism, and immunology. Phil thought that was a great idea and gave us a gift to get this center started. That led to a formation of a company as we got closer to a drug. And none of this could be done without all the people that work in the labs with us. So Dave mentioned a lot about the brain. And no one in their right mind would stand up here and tell you that the brain wasn't important, because then you wouldn't pay attention to the rest of the talk. Certainly important. But let me tell you, there is another part of the nervous system that you're also familiar with. Every time you touch something hot and you push back, that's your peripheral nervous system. If your stomach doesn't feel exactly right, that's your enteric nervous system. Many, many different senses, numbness, pain of all sides, comes from the peripheral nervous system, and that's these long axons extending all the way down, and we're going to talk about those. And Aaron and I call this a giraffe problem. So Dave mentioned that some of those axons can be really long. Just think about a poor giraffe. So axons can run all the way from their neck all the way down to their hoof. So that's probably 20, 30 feet. That's a very delicate, large structure, very complicated structure that requires many, many different protein products, and is also very fragile. And so that's what we're going to talk about. It leads to a lot of problems because of its fragility. And what matters in the nervous system is connections. Just like we try, and Dave and I have talked about how we collaborate, how geneticists and neurologists come together to make new findings. That's all about connections, right? That's personal connections. The nervous system acts the same way. It's about connections. So these wires, you wouldn't be able to make a phone call without wires, of course. Well, now you can. You couldn't. But these connections are incredibly important. And most of all, people have studied cell death in neurodegeneration, as Dave said. In Alzheimer's, we've always thought about these neurons dying. And now we realize that if the axons that make these connections are faulty and don't operate properly or degenerate for various reasons, that then you lose all continuity in the nervous system. And the brain can kind of be thought of as a central processor, right? And so it's kind of like if you don't have the peripheral nervous system to bring information to it, it's really kind of useless. It's sort of like having a computer that's not connected to the internet. So you really need that information flow back and forth, and these long axons are really the most vulnerable part of what we're going to talk about. So what basically those diseases are called are peripheral neuropathies, and they come from various sources. Diabetes. Diabetes accounts for a large percentage of it. Probably 30% of diabetics have neuropathy. It comes from chemotherapy, especially several of the chemotherapies, vincristine, Taxol that's used for breast cancer. Up to 30 to 50% of patients that are delivered those drugs have neuropathy. Pain, numbness, tingling that can last long after the therapy is no longer delivered. It can also be a problem because many times the therapy that's improving your cancer survival has to be discontinued because the pain is just too great. The nervous system is decomposing faster than the tumor. So if we need to fix this problem. And there's a lot of individuals. It's thought that by 2050 there could be 500 million people on the planet with some sort of neuropathy, largely being driven by the increase in diabetes in China and India and places with large populations. But there are many people out there that say they have cures. My mother had peripheral neuropathy for many years before she passed away. And even though she knew I was working on it, and she kept asking me, well, don't you have a cure yet? Don't you have a cure yet? Every week. Yeah. But your mother, so you have to. But eventually she said, well, I'm going to go to this neuropathy center. She lived down in Arkansas, and I hope, Arkansas is a great place. Medical care once in a while, not so much. So this neuropathy center was on Horse Barn Road, which probably tells you something. And they had advanced laser treatment for neuropathy. I don't know what they were lasering, but we don't treat the cause. We treat the cause, not just the symptom. And this is about a 10-year-old ad, and we didn't know the cause, and that's what I spent my life doing. So it's really interesting how, but people get roped in, but these pictures depict fairly well, these are their advertising pictures, what people complain about. Burning sensations, like pins pricking them in the feet, and it usually does start in the feet, and then eventually will go to your hands. But this is a major problem. People over, like Dave said, it's, I hate to say this, but you know, one of the biggest risk factors for neuropathy is, again, age, old age, older age, and I'm getting closer and closer to that time when many, many people will get neuropathy for unknown reasons besides just diabetes or chemotherapy. So what does it look like? What do we mean when these axons are damaged? Well, here's an example, this happens to be a mouse, but you see that, see here, where's my pointer? Oh, here we go. So these are axons basically lit up with a scientific trick. And you can see this is what normal looks like. So these axons are just long, slender threads. After damage, injury, or chemotherapy, they begin to fragment. And you can see that they're no longer continuous. They can no longer send a signal from one end to the other. Now, in disease, you're not going to have all of your axons look like this. There's going to be one checking out every week or every month, depending on the progression of the disease. But this is what happens. A discontinuity no longer can send a signal. So what did we know about that? Well, we knew only a couple things from some mouse models. We knew that there were ways that we could prevent this. And it appeared that the axon, when it was sick or injured, knew that it should degenerate. There was a self-destructive process. And the only clue we had about that was that a molecule called NAD, which is the prime energy source of life in all species on our planet, seemed to be involved, that it would get degraded, and it was sort of like the best way to check out if you're trying to turn off a light bulb is just to pull the plug and lose the energy. And that's essentially what the axon was doing when it thought it was damaged and should go away. So we wanted to understand more about that than just that it was an energy problem. So we did what's called a genetic screen. So we took neurons from mice and put them in a laboratory culture, and you see they make nice axons even when you're growing them in the laboratory. Now what we wanted to try and find were any genes that would prevent an axon from activating that self-destructive program, and even though it was injured, it would still remain intact. That's what we wanted to try and do. So we decided to screen every single gene in our genome. That's 20,000. So you need robots for that. So here were our robots. Yo and Josiah, and they worked on that for at least a year until they came to me and said, we're really work hard, but we're not going to do this robotic stuff anymore. So we had to buy some robots and do it this way. And here, at the end of the day, out of all 20,000, we found one gene that really was miraculous, and I'll tell you about that. But here is what we had to go through. When we moved labs, I'm kind of a pack rat, and we kept all this just as evidence of how hard we work. And when we moved labs, we had to ship it out and throw it away. But this is the detritus of all those screens, hundreds of thousands of tubes in those boxes. What we found was a molecule called SARM1, and we found that that was the central driver of axonal degeneration from any cause. Chemotherapy, different types of neurodegenerative diseases, genetic causes, mechanical injury, traumatic brain injury, anything. It is a central program. There are many ways to feed into that funnel of a central program, but it is the central driver of the execution of the dismantling and degeneration of the axon. If you remove it genetically, it prevents axon degeneration. Mice, where we manipulated mice so that they no longer have this gene, if you give them chemotherapy, they do not develop neuropathy. They have no symptoms of neuropathy whatsoever. They don't feel any excess pain. They don't have any itching, none of the things that you might think about for that. So how does this molecule work? Well, here's just a cartoon, and you just look at the yellow boxes. So here's the molecule, and the green and yellow boxes like to interact with each other, and they hold each other together. As long as the two yellow boxes don't come together, your axons are fine. But in disease or an injury, this interaction between green and yellow falls apart, and the two yellow boxes come together. And when they come together, you lose all your NAD, you lose all your energy, and the axon degenerates. So that's the central scheme of axon degeneration in most diseases. And here's what it looks like just in pictorial form. So these are axons grown from neurons grown in the laboratory. That's what healthy looks like, nice continuous lines as you saw before. This is what happens if you injure them in the presence, in a normal genome, when you have a SARM protein, which we all do. That's what happens. If you take away SARM, even though you injure them, they look perfectly intact. And the same thing happens in a mouse nerve. This is after you injure the mouse nerve normally, and this is if you do that in one of these genetic mutants. The reason I show this, because it's quite dramatic, but also you realize that if you could inhibit this molecule, you could probably have a drug that would stop this process, because if you can just eliminate the molecule, you stop the process. So that's the idea. How can we block the activity of this protein? And so we did that. We discovered that SARM is an enzyme, and it actually is the enzyme that cleaves your NAD. So normally in all of us right now, hopefully, that enzyme is quiescent in our axons. It's not really doing anything. It's held in check by some process we don't quite understand yet. But once there's injury or disease, it becomes activated and begins to chew up all of the energy source. Enzymes are great drug targets. They're the easiest thing to target. So once we found that, that was sort of a breakthrough, and we're developing now first-in-class treatments to protect axons in a variety of neurological diseases, which is why we had to start a company as well. But in our initial drug screen, we immediately identified some inhibitors, and we tested those, of course. And so here again are those same sort of axons. These are axons after injury, and these are axons after injury in the presence of the drug. So you can see they look pretty good. They're not as good as the genetic mutant, but this is the first example of a drug that could block this protein and therefore protect axons from degeneration. So you can see where we're going with this. As we develop better and better drugs, we'll be giving those to mice, then we'll be giving those to people, hopefully. So life is a balancing act, and NAD here is the energy source I mentioned. It's the balancing. There is another enzyme that's the guardian angel of the axon. So this actually makes NAD. So normally you're making plenty of this enzyme, and you're making plenty of NAD, and everything's fine. And then activated SARM is the devil, and it's going to chew up all the NAD. So life is a balance. What happens in people that might have mutations where that balance is disrupted? So human genetics highlights SARM's role in disease. So there was a neurodiagnostic mystery, and this is it's a sad but interesting story. So there were two brothers. At about 1 year of age, they both had trouble walking. One of them actually developed it about at age 2. He walked fine and then began to decline and stopped walking. His younger brother had a more severe form even and never did walk. They developed sort of like a Guillain-Barré-like syndrome, where you might be familiar with that after polio, where you become suddenly paralyzed over a 1 to 2 or 3 week period, sometimes have to be put on a ventilator, and that's usually an immunologic attack on the nervous system. They thought that's what this was, even though Guillain-Barré is extremely rare in this age patient. But they treated it as if it was Guillain-Barré with steroids and doing some plasmapheresis, and they got better. Then they relapsed. Then they realized they were dealing with some other kind of malady. And they thought it was some sort of immune demyelinating neuropathy. It was a really diagnostic mystery. This was at UPenn, our collaborator, this patient was at UPenn. She's followed them for more than 20 years now, and they have a relapsing, remitting course that's almost like multiple sclerosis, but it's in the periphery, it's not in the brain. They'll get better, then they get very bad. When they get very bad, they have to go into the hospital because they will actually become paralyzed and have to be put on a ventilator. They give them steroids for a couple of weeks, they gradually recover, but never quite to the level they were, so very much like chronic progressive MS. They'll lose all muscle activity during that time and then will gradually recover. Now they're developing atrophy in their optic nerve, so in the CNS. They're gradually losing their eyesight. They have episodes, when they're not being paralyzed, they have episodes of severe burning. Their skin turns red, their feet and hands swell dramatically. And yet, there's no cognitive impairment. The one in an incredible display of perseverance and mind over matter. One of these children is actually in college. He's been wheelchair bound since he was 2 years of age, with all of these symptoms, having to go into the hospital and being put on a ventilator and then trying to go to college. It's amazing. So the mystery was solved by analyzing their genome. DNA sequencing revealed that they have mutations in that guardian angel, NMNAT2 enzyme. So they're not making enough NAD in their axons. They don't have a counterbalance, and so this balancing act is out of whack. So mutations lead to this robust activity of this SARM1 thing that should not be on all the time, but it's on all the time in their neurons, so they degenerate. And we hope to be able to help. Now, it'll take a while to make a good drug, but we got lucky and found another source. And Dave mentioned to you that gene therapy for neurodegenerative diseases is becoming not only a pipe dream, but a reality. And in fact, the slide that Dave showed on SMA, which is the gene therapy type of treatment, the first author on that is Rick Finkel, and he was a classmate of mine here at Washington University. So WashU does a really good job of trying to train people to help people with neurological diseases. So our therapy, we got lucky, and we were doing what scientists do, fiddling around, trying to understand how this worked, how this SARM molecule worked. And we made a mutation, a bizarre mutation, that enabled us then to come up with a gene therapy approach. So if you look at the diagram we had before, you realize that this interaction here, I told you when you injure, this interaction falls apart and these two yellow boxes come together. In the mutation that we made, this never falls apart. It's too sticky, so that it never gets activated. But even better than that, when you put this into a normal cell, it goes in and binds up all the normal SARM so that it can't get activated either. So if you put that into a cell, the axon doesn't degenerate. If you put that into a mouse and injure its nerve, those nerves remain perfectly intact. The mouse does not know that its nerve has been injured. It's perfectly fine. So we think that this is a novel way that would be actually a more direct way to impact these diseases. And especially the first one would be one where we really understand the mechanism and is in dire need of an experimental therapy. So Dave and I have worked together for a long time, and you can see the necessity of understanding these neurodegenerative diseases. And there are great clinics here for the ADRC, for Alzheimer's. There are clinics. He mentioned that Tim wants to start one for ALS. Peripheral neuropathy actually is coming in from multiple sources. So for chemotherapy-induced neuropathy, one of the major things for the Siteman Cancer Center and cancer centers throughout the country now, as people are cured of their cancers, cancer survivorship, cancer rehabilitation, cancer patient rehabilitation from the therapies that they endured is becoming an increasingly powerful topic. It's a very important topic when you're trying to, we think that things like this can really make Siteman number one. It's number 3 now, but the idea that we can prevail over many of the sequelae, if we could develop treatments for this. So we want to start a neuropathy clinic. Why? It's the most common neurodegenerative condition. New treatments are on the horizon and they're critical, and the time is now. And the lessons learned from neuropathy about axonal degeneration can be applied to multiple diseases, Parkinson's, MS, maybe Alzheimer's, etc. What do we want to do? We want to develop a clinic where we can study patients with unknown genetic causes, perhaps, of neuropathy, those receiving chemotherapy at Siteman, those followed for their diabetes. We have huge endocrine clinics here. And patients with related disorders. We want to understand we want to bring them together so that they can get proper care and that also they will be available for genetic analysis and stem cell therapies, etc. How? So we want to combine fantastic patient care with genomic studies, some of the new technologies I'm going to tell you about just briefly, and novel molecular assays that will help us identify even more drug targets. But you need to have a good feel for the natural history of the disease. So as we learned, unbelievably, that right now, chemotherapy-induced neuropathy, there are no natural history studies. No one knows how long it takes to get the neuropathy. No one has a biomarker. Dave talked about biomarkers. There's no biomarker for this sort of a neuropathy either, but there is something called neurofilament light chain, but no one knows how it correlates with the therapy itself. Do you give the therapy and then check the biomarker in the serum? How do we study this? How do we do clinical trials in this? We need a clinic that we can do these sorts of things, more genomic studies, et cetera. Why here? Why now? It comes back. It comes back to connections, the connections that we have as colleagues at Washington University, where people as different as geneticists and neurologists and surgeons can work together to make new therapies. So we have colleagues in all these areas. We've reached a turning point in our understanding of these conditions, and we can be first. I mean, in translational, because of the Hope Center, in translational neuroscience, WashU is at the top of the game, and we want to remain world leaders in all of these disorders. So as I mentioned, it's important to have the patients and follow what's going on with them so that we can deliver better care and hopefully cures. But new technologies have also opened up new windows of opportunity. So at the Mcdonald, at the Mcdonald Genome Center, obviously we sequence DNA. I'll give you a couple facts, fun facts. So this sequencer right here called a NovaSeq, $1 million a piece. Well, what does it do? We can sequence the genome of 1,000 people per month on this machine. So in 2005, wasn't that long ago, at the Mcdonald Genome Center, it took 250 years worth of time to do what we can do in one day on this machine. So the idea of sequencing a genome was a many-year project. And as I mentioned, 1,000 genomes a month from one machine to find the genetic causes of these diseases. And we want to compare those genetic causes with the phenotype. Here's a good-looking neuron. That's hopefully a sick one. But we don't want to go through this again. And now all of this has been reduced to a single tube and a single plate like this, with the help of new instrumentation like this. So this is called a Phoenix. It's also $1 million baby. But what it will allow us to do, if we can find one, get one, is it allows you to take tens of thousands of pictures of cells per hour in multicolors. So that, and this is the beautiful pictures that it takes, allowing you to then use artificial intelligence to find out which cell is mutant because of its phenotype looks different than the others. So that's what we want to do. And these kinds of machines speed things up from a 4-year process in all these boxes to single plates and pictures like this using computer technology. I just want to give you, once we identify an important cell that we think is mutant that looks funny, then we use an instrument that looks like this. And I just want to show you a short movie. It's not like Gone with the Wind, believe me, but it's kind of cool. So here's, you're growing the cells on this plate here, and it'll turn in just a minute so you can see it. And they're growing on a matrix that has a little bit of magnetic material in it. So here they are. And that green one is the one I want to get. That's the cell of interest, as it says there. Okay? So now, up from the bottom, it pokes up that cell. And then this magic wand comes down. It's got a magnet in it. Picks up that magnetic thing that it's growing on and drops it in the tube for you. Now, it'll do that about 1,000 times an hour. So you can collect many, many cells, and you don't need Josiah and Yo to sit there all day long and do that. So these are the sorts of tools that allow us to begin to utilize genomic information, get to cell phenotype, and then to drug screening to identify cures. So I want to end by saying that this is a new era. The technologies are more powerful than we ever dreamed, or at least than I ever dreamed. It gives us unlimited potential to help people. Here's a little girl, as Dave mentioned, with SMA. As Dave mentioned, most of these poor children die within the first 1 or 2 years of life. She's 3 years old and walking around. This is a little boy that has Duchenne's muscular dystrophy. He's riding a bike. And the question now is, clinical trials are ongoing that we'll be able to give sight back to people through these genetic technologies. So really, being able to, save a little girl's life, be able to give sight back to someone who's going to go blind, these are times that are really extraordinary times to be a scientist and a clinician, and the tools are available to help us. So thank you for your attention.
We definitely have time for some questions from anybody.
Yeah, sure.
Yes.
I got one for each of you. So what, because you're looking at peripheral nerve what about chronic itch? Where does that fit in? Because it's not necessarily pain, but it's still a peripheral nerve issue. And then also for Dr. Holtzman, what about the new utilization of eye exams for detection of early onset Alzheimer's?
You want to start your first?
I can start with itch. So Washington University is unique in that it has the only center for itch in the United States, or maybe in the world. So there are people studying itch as a modality, and there are new treatments on the horizon. In fact, one is in clinical trials now that looks fantastic. It's a drug that was originally developed for cancer. It's called a JAK1 inhibitor. So itch is a very close cousin to pain. As you know, if you itch something long enough, it'll get pain and actually relieves the itch, and that's almost better. But they've identified the circuits of what's going on in itch. They know exactly what receptors, what neurons are involved. And so that has gone from a backwater of science where we didn't understand anything to a rapidly progressive field with novel new therapies.
So in regard to the detection of Alzheimer's disease before it is obvious clinically with an eye test, so there's sort of three things that are very promising. Some we already know work, and then the eye test. So one is measuring the accumulation of these proteins with amyloid and tau with PET scans. That can now be done. It's very expensive, but it works. So you can detect the disease before somebody develops memory loss. There's also recently, my colleague Randy Bateman at Washington University developed a blood test that can detect the buildup of abnormal amyloid in the brain. That appears to work, and I suspect over the next few years, several companies will have tests that can detect Alzheimer's disease in people when they're still normal. And then this third thing is the eye test, also recently published from Washington University from the Department of Ophthalmology, where just as a bedside test, you can look at the thickness of the retina by looking through the pupil. And they detected in a relatively small group of people that patients that are already developing amyloid deposition while they're still normal have thinning of the retina. So what needs to happen next is that needs to be validated in larger numbers of people, but if that's true, it might serve as a nice diagnostic test that can be used. It's very simple.
You mentioned gene therapy a few times. How is it different in terms of as a form of treatment? How is it different than some of the other treatments that you described? And as it relates to drug approval, compassionate use, this accelerated, the FDA seems to be, in some cases, approving on a more accelerated basis than others. What's your view on that, and especially in these conditions where there's little hope? You mentioned the young boy on the bike with Duchenne. I know there's a controversial drug that was approved for that. What's your view on those accelerated treatments?
Well, gene therapy has gone, well, if you want to talk about orphan drug treatments, go to gene therapy for a minute. It's gone from, a pipe dream, and in the last 10 years has become mainstream. So there are many, many treatments right now being approached. This has largely been driven by the discovery of what's called adeno-associated virus vectors. They're very good. They seem to be very harmless so far. They don't have you mount an immune response, and I think that's why this is really being driven. They can be made so that they're tropic for the nervous system, especially various specific cells. It's a cottage industry now to find all kinds of different AAVs that will deliver the payload only to a particular cell type. So I think if you go through the clinical trials now and things that are in the pipeline, there are literally hundreds of new gene therapies for many types of diseases. People like to use gene therapy initially on an orphan disease for exactly some of the reasons you mentioned, that these are conditions that have no other alternatives. And so that's the first place that people are checking them. SMA would be an example of that, a rare, thank goodness, a very rare disorder where it works. And it will be moved into more mainstream disorders as those trials progress.
Yeah, the controversial treatment for Duchenne's that you were mentioning, there's a treatment that really didn't look like it had too much of an effect, but more recently, which is not still yet approved, just last year, new data was shown using these adeno-associated viral vectors to deliver dystrophin, which is the mutant gene in most common form of muscular dystrophy, where they express that protein using this viral vector throughout all the muscles in the body with a single injection. And in the early data with just a few patients, kids that should be degenerating are actually getting better. So there's now a big trial that's being initiated to really prove whether that is true or not, but it looks really promising.
Have you done any studies about the degradative effects of pharmaceuticals on cognitive function? As you mentioned, side effects of cancer treatments can produce negative.
Yeah, so you're talking about trying to use it as a neuroprotective for the ravages of chemotherapy, especially on children, or chemo brain, so to speak. Is that what you're referring to? Is that what you meant?
No.
No.
Maybe clarify.
Okay. Can you clarify it?
Yeah.
I'll talk to you later.
Okay.
Okay.
Any other questions?
If there's one more,
Yeah.
otherwise we'll break. And thank you so much. Let's thank our presenters. What a rare treat to really be on the leading edge, in the forefront, and the inside of all this information. Thank you so much for all that you do. Thank you all for coming. They have agreed to stay, so we have time and space for you to ask more questions and follow up on questions. And we have a good another 20 minutes or so that you can ask questions of them behind. My closing commercial is to follow the foundation on Facebook. Come to our website. We would love to hear from you. Thank you very much for coming this evening. Bye.